Hello, welcome to the Seminar Method. I'm Flora Honoré, and today I wanted to present you the Pitchum team. Pitchum is for Project Engineering and Technology for Universe of Microbe. Why did we create Pitchum? So the idea is to open access to technology that are difficult to access, and we know that it takes a lot of energy to get started. So for that you need to be trained, and you need specific equipment for different technologies. And those technologies are the flow cytometry and microphysics. So why we have created an hybrid lab? So the idea is to have continuous feedback between researchers and the platform. So we have two different axes of research, one on flow cytometry and the other one in microphysics. And in the middle, you have the platform. So this is a team organization. You have Gaël, Christophe, Robaglia, Loïc. Me, I am on the platform service. And in research in microphysics, you have Nicolas, Jean-Raphaël, Madi, and Satish. Jean-Raphaël will be also on the platform service, but only on microphysics. So I will start with microphysics. So you may already have this kind of experiments. If you want to observe your bacterial cells, the most simple option is to look at on a solid surface, for example on agar plate. And for that, when you observe the cell under an optical microscope, you will see that the cell adhere to the surface. You have monolayer, and you can do live imaging. But modify the medium will be complicated. If you go now on liquid, in liquid you will do most of the time between a slide and a cover slip, and you will have this kind of pattern. So no cell adhesion, no monolayer. Live imaging will be difficult, and also modify the medium. So innovation was needed in microscopy for bacterial imaging. So we propose two different options. The first one is a chemical adhesion. So the idea is to make adhere cells on chitosan, and the chitosan has been developed by the TAM-Minus team, and now it is commercialized by IDIL. The other option is cell ASIC-like, which is a physical confinement. So the cells are confined by a ceiling, and we can use it for various organisms. When we have a new microphysics project, we will go this way. So we first try with the simplest chip. If it works, we stay with it. If it don't work, we go to a more complex chip. And the simplest option is a chemical adhesion. A more complex chip will be the physical confinement. So the simplest microphysics we use the chitosan, and the chitosan is obtained by the deacetylation of the chitin. And the chitin is the second most abundant biopolymer present on Earth. So you can find chitin on insects, crustaceans, fungi, and algae. If you look without chitosan, for example, here you have E. coli cells, you won't have any adhesion of the cells. But in presence of chitosan, you will see that cell can grow, and they are they adhere to the surface. Why we said that it is the simplest option? Because it is really simple to use. So IDIL commercialized the chitosan coated slide. We stick on the chitosan slide an EBD channel, then we hydrate, and we can inject the cell directly into the channel, and we can centrifuge, and this step is optional. I wanted to show you now some example with the chemical adhesion. So this is the list of the different bacteria we can use on Ketozen. It is a non-exhaustive list, so we're just starting to discover which bacteria we can use on Ketozen. And at the beginning, we were thinking that Ketozen can be used only to immobilize the cells. In fact, we can also follow cell motility, for example, with Myxococcus xanthus. And we have seen that we can also use it with Acanthamoeba castellani. And this experiment has been done with Laurent Bouteille from the IGS lab. We can rapidly change the environment for drug study, for example. So here we have E. coli Tolbe M. cherry strain. We can inject the CCCP directly into the channel, and you can see that the CCCP induces depolarization of membranes, and we can see the septal localization of Tolbe. We can also do a statistical experiments where we can follow the cell growth in presence or absence of colicin A. You can see that with colicin A, the cells don't grow, but in absence of colicin A, they can grow. Since I work at the beginning in Time team, I could not resist to observe predation assay. So I did the predation assay on Ketozen, and you can see that when we inject Myxoxanthus cells inside, and you have E. coli here in red, you can see that cells start to spread on the surface and they go to kill E. coli strain. You can see on the zoom that some cells are killed by Myxoxanthus cells. Imagine now you can stop this event by fixation and observe directly at the biological process at the molecular level. It is possible because we can do immunolabeling. So the principle is to first observe the cells, for example here on predation, capture the event by fixation, and then stain with a specific antibody. The first one will target your protein of interest, and the second one with the fluorophore. And we choose to show you an example with pill A, which is a major pillin of the type 4 pilus, and the fluorophore is an Alexa fluor. You can see that we can nicely label Myxococcus xanthus, and we can also see some polymerized pill A, which are in action, so they allow the bacteria to move, and we have freeze this event. Now I wanted to show you an example of a new Pitchum project. So Sibyl Talchamp and Eva Vanaria from the LCB wanted to observe Lactococcus cremoris cells during different aerobicis conditions. So they use a CyB GFP strain. CyB is a cytochrome which will have different expression in different conditions of oxygen. And so they wonder at what oxygen concentration expression of CyB occur. So we first try with the simplest option, which is the Ketozen. And you can see that clearly the cell have difficulty to adhere to the Ketozen. And once some have adhere, they will start to divide, and they will detach from the surface. So this is a current problem we have with gram-positive bacterium. So we went to the more complex chip, which is the one with the physical confinement, and we call it Mixat. So this is how it looks. In the center of the chip, we have an area of interest, and in this area of interest, you have a ceiling which is supported by some pillar. And you can see that the pillars are here in white and here in black. So you can see how big is this area of interest. And so we have inject Lactococcus cremoris directly inside the channel. And so the little dots are the cells. So we have observed Lactococcus cremoris under the ceiling, and you can see that they managed to go under the ceiling, but still some cells can move. So we choose to have to go with another option, which is a more complex chip, and so we will increment also the O2 control. So we have done the co-development with Jean-Raphaël, Nicolas, and Igor Ozrov from the CNM. They have developed stairs, which is actually an inverted stair ceiling. So the cell will be blocked in function of their thickness. So this is what it looks. So you have different steps, and so the cells are injected from here, and they will enter into a funnel, and so they will be blocked once they cannot move. During the Pinus cursus, we have done last week this experiment to show that we can actually use different E. coli cells with different lengths and different colors. And so you can see that the cell can enter, the smallest cell can enter here, and the biggest one will stay here. And we can also see the cell growth. So if we come back to our new project, we can see that we can now clearly immobilize the cells under these new chip stairs. So we just have now to do the same experiment, but with O2 control. Okay, so now we have stairs. It is suitable for most of IMM organisms, and the goal is to have universal system, so we can make other stairs with different height of steps, and we can increment some options as gas control, chemical gradients, and porous environments. So just to show you some example of physical confinement, we can use it at a single cell level, for example for E. coli strain here, for cyanobacteria, and for fungi. At the single cell level, we can also follow the dynamic of phage infection in E. coli. For example, for Laetitia Rio and Roman Guarino from the list, we use the physical confinement mixate, and we have seen that for E. coli cells, we can infect them with filamentous phage, and you can see here on the movie that the little green dots are the phages which are entering inside the E. coli cells. For the project of Nicolas Ginet and Judith Sarr from the LCB, we have used stairs, and we can see some nice phages, which are capsid phages, turning around E. coli cell here. For the Claudia Petrios project from the LCB, we have managed to grow the fungi directly inside the chip. You can see that so the chip is dry, and we fill the chip, and then we inject Myxococcus xanthus cells, and now we can observe the interaction between the fungi and the bacteria. Imagine now you are on the battlefield, and we wanted to follow bacteria-bacteria interaction, and so we use E. coli here in red and Myxococcus xanthus, and we wanted to know which one will win this little war, since E. coli can grow very fast, and Myxococcus xanthus is able to kill E. coli cells. And so you can see that for now we don't know which one will win. If we zoom in this area, you can see that Myxococcus xanthus clearly go to the contact to E. coli cells and kill them. and so they will won this little war. And so this is the kind of thing we can do in microfluidics for now. Okay, so now I'm done with the science of the microfluidic part, and I will talk about the platform service overview. So if you have, so this is how it works. If you have a new project, you can ask for to do the experiment as a service. So I will do the experiments. If you want a co-development, it is possible. It is for the more complex projects, and we will need a continuous feedback between the researcher from the team and you. And you can also ask to do it yourself, so you will use the platform facility, and you need for that to be trained. So last week we did the Plinus Cursus. It was mostly for the PhD and master students. So it was a real success since we have developed the stairs for this Plinus. This is the workflow we use for code development. So you have different steps. The first one is the concept and the design. So you can ask for that to Nicolas. The other one is the clean home part. So we use the clean home, the platform planet from the CNAM. And so you have different steps, the lithography mask and the photolithography. The first one will help you will obtain the mask, and the other one you will obtain the wafer, which is actually a mold. For the soft lithography step, so it is done in the LCB in a grey room. For that, you can ask Nicolas or Jean-Raphaël. They will do PDMS devices, so it is commonly the chip we will use for the next step. Once you have your PDMS device, we just have to do the abiotic test, which is without any microorganism. And this is done to make sure that the device is working, and so we can use it for the next step. Once you are sure that the chip is working, we just have to do the observation with a microorganism. So if it is in case on the platform service, I will lead the experiment, and we will also do a live imaging. So I'm done for microfluidics. Now we'll talk about flow cytometry. So with flow cytometry, you will have a statistical view of a population, and it will allow you to look at the variability in a population. And this is complementary for the microscopy. Since last year, we have the Cytek CS spectral sorter, which has been obtained by the IMM, but it's difficult for the community to use it. So I have started to be trained by Gael on pilot projects, and the idea is to be operational before the end of the year. Now, this is the platform workflow. So if you have a new flow cytometry or cell sorter project, if it is a long-term and collaborative work, you will still working with a researcher. If it is one-shot experiment, so I will lead the experiment with the supervision of Gael. So we have a high-performance flow cytometer. We have 5 different lasers, so we can use different fluorochrome and detect more markers simultaneously. For the cell sorting, we have 6 different ways to sort, and we can adapt the sorting to different cell types. And for now, it is only for non-pathogenic bacteria organism. And if you want to do the experiment in flow cytometry with your pathogenic organism, you will have to fix first before to do the experiment. So just to show you some example of application in microbiology. So with the flow cytometry, you can evaluate cell viability. for example, with antibiotics. You can also follow the gene expression using fluorescent protein to see, for example, the heterogeneity. You can follow the cell cycle. You can quantify the cells and do biofilm analysis. You can also differentiate microbial population. For the cell sorting, you can do isolation of specific microbial population and select genetically modified cells, all of that based on fluorescence. You can enrich your population, for example, if you want to look at persisters. You can do virus and phage host interaction studies if you want to isolate specific cells which are not infected, for example. And you can also do microbial ecology studies. So we are opening a call for projects in flow cytometry and cell sorting. So the deadline is June 1st, and you can scan the QR code or contact directly me on with my address. And you can also go to the website. What to come next? So the website is in progress, and we also need to be a physical team since we are now spread on the LCB lab. And for the training, a new Plinus courses in flow cytometry is open, so don't hesitate to postulate, and it will be in June 10 and 11, and it will be done by Gael. So how much it costs? For now, the philosophy of the platform is to align costs with real usage, so the service is free for the first year, but consumable have a cost, so you will pay only for consumable. So those are the current and future projects of the platform. You can see that we have a lot of microfluidic projects. We have some in the LCB, but other from the LISP, the BIP, and the Pasteur Institute. And in flow cytometry, we have only a few ones. So don't hesitate to postulate, and I hope there will be more to come.